welcome Miss Mala on the stage and Thomas also on the stage. And they don't need any introduction because we already introduced to all the audience as well. No. So we will start with the session where I will just pop the questions from the follow-up questions that we have already got for them. So first question goes to Thomas, then the question goes to Mala. Then I will pick your question in the set of questions that we've already received just while you were talking.
Oh, okay. I got a lot of those.
Yes, yes. So we will just try to address most of them. In case
Okay.
if we do not get the time to get around all the questions, then we can ensure that we probably send you those questions by email if that is okay with you.
Yeah, not a problem. We can do white paper. Like I said, I like to do a white paper, and I did it for the Brazilian ISPE, Malaysia, and Indonesia. It's the same thing. They sent me a bunch of questions, and I wrote a white paper, and I addressed all those.
Okay. All right. That's great. Okay.
So
So
putting
we
things to paper and words is totally different approach, right?
I agree with you.
Hey,
I
Thomas, nice to see you.
Very good morning. Hi.
Good morning. What
I
time is
think
it
we
there
have Mala
for
as
you?
well.
Where
Mala.
are you at, Thomas?
I'm in my home office in Germany, so we have just like 8 o'clock in the morning.
Oh, okay. So, I'm on ASTM committee, and it's usually, for me, some of those morning sessions or thing. It's almost what time here? It's almost 11 o'clock here. And I was
Okay.
up at 4 a.m. this morning, so I'm just running on iced tea petrol right now.
Right. Okay. Okay. So, welcome Mala, welcome Thomas, and we
Hi,
have
Mala. Yeah.
David. Means you already attended his session as well. So, what we're gonna do is that we will just ask the questions in order. Like first question to Thomas, then Mansi will be putting in the second question that we have got for Mala. Then we will pick the question for David after that. So we will go in this order. So the first question that we have for you, Thomas, is during PDE calculation. If LOAL is not available, then using NOAL is acceptable. What are the regulatory aspects on this?
Yeah. So this is really something where we say there's no observed adverse effect level. What David also just outlined is really the major part of the PDE calculation. So that we have this no observed adverse effect level, and this is even outlined in the Annex 15 that this kind of health-based exposure limit calculation can be based on the no observed adverse effect level. So that would be the perfect approach to do the calculation on health-based exposure limits.
Thank you, Thomas. And next question we have is for Mala. And at the time of the development of cleaning validation, our MACO came to 0.12 ppm, and our LOD was 0.09 ppm. After some time, we added some equipment to the equipment train, and after adding the revised MACO was 0.08 ppm. So the question continues, I guess. Mansi, can you put the remaining question? It's only half the question. Can you put the full question? So I guess, Mansi, can you put the remaining question, please? We're waiting for you. Okay. So the
I
complete
think I
question.
think I think it says something like which
What should
means
we do? Means it just wanted to say what should we do? That's the question, Mala.
yeah.
Sorry.
Yeah. So good morning, all. Well, if your revised BACO, like after your equipment addition, if it is going to a lower level than the validated limit, what you could do is that you can revisit first your method, whether it's a HPLC method or a UV method. The second thing that you can also do is sometimes the methods get developed at very early stages, and you can see whether any improvisation can happen in the method, and you can achieve a lower LOD. Okay. If that is not possible, this is what I've given the answer as. So you can read it on the screen. And you can also review to extract the swab with a lesser quantity of solvent. Okay, so this helps in enhancing your visual limit, and you can see that. And also we can do one more thing is that you can sample a larger surface area, which can be 9 square inch or a 16 square inch. Okay, that is another option which we can do it. And the last thing is that you can also enhance your batch size of the next product. Okay. If none of these are possible, the last thing what you can do is switch over to a dedicated equipment tray.
Thank you. Thank you very much, Mala. And
I would I would also like to add that I would readdress your macro calculations too.
Yeah.
See, I would readdress those based on those suggestions that you gave because those are really good suggestions. But I would look at those too.
thank you very much, David.
Yeah.
David, the question that we have for you is by Mr. Singh from Beatrice. What would be the acceptance limit for DEHT study? Is there any rationale or guidance for that limit?
It's not 3 years, like I said before, but you gotta. It will vary. Your dirty old times will vary dependent on all they state is, as I mentioned before, what you're going to look at is when you have a dirty old time, it's going to be dependent on the material that you have. Okay, and every material is different, composition, solubility, and that. So you have to do it case by case. So I typically how we do dirty old times is we do development studies and we do time intervals where we pull samples of the equipment and then we establish a par, which is a proven acceptable range. So, for example, if I pull on day 0, day 2, day 3, day 4, day 5, day 6, I'm going to pull at time intervals. And where I get the failure, I'm going to put a safety factor in, and I may go back maybe 2 days back to ensure that I never exceed those limits. So that's how you do it. But you'll get a guidance document. As I mentioned before, if you get the ISPE document, it does describe in detail how to perform those tests and what samples to pull in that, because you're looking at microbial, you're going to be looking at degradants at that time too, and you're going to be looking at what transformation has happened to that product, because it could be a totally different product at that stage, right? And that if you leave something there long enough, which influences your cleaning process. So all these things are interconnected. So you got to look at that from a risk perspective. Okay.
Thank you, David. And Thomas, the next question that we have is for you. Um, should we go for CEHT studies after introducing a cleaning agent as a kappa?
Uh, what should be the CEHT? Cleaning estimated hold time, maybe? Is
No,
that
it's clean equipment old times. So,
clean equipment hold time?
yeah,
Okay.
yeah.
Mm-hmm. Very
Yeah.
good. Okay, so if we have this kind of issue that we have like corrective action, preventive action done, or and introduce the cleaning agent, we are optimizing the situation of the clean status. But if you have the clean hold time already evaluated, so your equipment will be standing in an environment what is controlled usually, and so these kind of circumstances will not change just due to the fact that you have used the cleaning agent. So I would outline that the use of the cleaning agent will bring you in a better way to the clean status of the equipment, but the clean hold time was evaluated just to identify that there is nothing happening with the clean equipment until the next production starts. So it's not really a need to go for that if you can show that this circumstances so that the clean status is achieved and that the clean room conditions around this equipment did not change. So there's no humidity more, or there's no introduction of water due to the cleaning agent or something like that. In this case, I would not redo the clean whole temp studies.
Yeah,
Thank you
good.
very much. Thank you so
Goodness,
much.
I just would like to add on that uh, what Thomas mentioned. So we need to also have a rationale why the detergent was added. Okay, as part of a kappa, there should be a rationale and risk assessment whether that also could suffice instead of having to repeat the entire cleaning validation again. The
Yeah.
Okay.
If,
Thank you.
if you're adding a new cleaning agent, then that can change the entire dynamics of
yeah,
your cleaning
Yes.
process, the residual limits and all that. So you need to do a risk assessment. And Thomas is right. You got to look at the initiation of the next subsequent manufacturing operation, whether it's sterilization or if you're just going on and going to reuse the equipment and that, or if there's another sanitization step. But you're absolutely correct.
Thank you,
and we,
David. Thank you, Mala. And Mala, I have a question for you as well here. It's from okay, Anand Mahajan. Question for Mala Mohan: how much is the area light LUX intensity requirement for visual inspection for verifying equipment?
well, it's a good question. So the light intensity, what I would suggest is it depends on many other factors. It's not only the light intensity, it also depends upon the angle of view and the distance of viewing. Okay, so that is very much important when you consider the light intensity. What I would suggest is minimum 200 lux will be good to have, and the other thing is that we can prepare solutions of the LOD concentration and view it under the different intensity of the light. Okay. So wherever you can view your minimum LOD concentration or LOQ concentration between say 100 to 500 lux, that you can consider as the light intensity for a product in which you can view the concentration.
Thank you very much. Thank you, Mala. And
Yeah,
additionally,
and I went on to add on that, whatever light source you use during validation, continuously use that. Don't change up. If you're using
Mhm.
one type of flashlight and you decide to go with another one, that may change your entire visual inspection and that. And I've seen that happen. I've seen companies get cited for that. So be consistent in that. Yeah.
okay. David, I have a question for you by Akshay Bhatt. Is there any regulatory guidance for challenging the DEHT in how many batches, whether one or three?
Um, I typically do three for dirty old times and that, so I can have a statistical value. And I've seen some companies, if they've done a good job during development, just do one and that. So it's based on what your risk is that you do. But one is only guessing. Two is giving you a little more degree of assurance. Three gives you much more higher degree of assurance. Okay. So just remember that whatever you decide to do, they're going to challenge it. So remember what I keep on going back to: scientific rationale and justifiable defense of what you've done. So if you can defend what you've done with objective evidence and that, then that's fine. But we do three runs, the dirty old runs. And I think most of the industry people that I've worked with do the same thing, unless somebody else does something else. I don't
Thank you. Thank you very much. Okay. So I have a
know.
question by Bhavin Shah from Sun Pharma for Thomas. Um, is analytical method validation required for products other than worst case products? So it seems to be this was after your session that he had put this question, and I'm
Mhm.
sharing it with you now.
Yeah, so the analytical method validation is usually needed for all the products what you want to analyze. But if you are talking about only the worst case identification, then I would say the worst case identification is done with an analytical method what needs to be validated in accordance to ICH guidelines. I
Yeah.
would always do any analysis in the terms of cleaning validation or even in the pharmaceutical manufacturing based on the ACH guidelines, because that gives you the reliable results what you really need. So I do not know if there is a fixed guideline on that to have only analytical methods available also, for example, for byproducts or for detergents. But we usually outline that the validated analytical method is a basic requirement for all analytics in pharma.
Yeah.
Thank you, Thomas. David, you have something to add?
No, I was just saying he's correct. I seen that. Like it depends on, you may have one methodology for a detergent and that, for example, conductivity and that. Um, but if you start seeing your cleaning agent as your worst case soiling, you have a problem there. Okay, so just be aware of that. So you try to standardize, and if you can get away with using one method, keep it simple, keep it practical, keep it achievable.
Thank
Let's.
you.
Yeah.
Thank you, David. Uh,
You're right.
the, the question that we have for Mala is what level of extraneous peak during swab analysis requires investigation?
Oh, yeah. So the extraneous peak, it's a good question. So the extraneous peak, what comes? We have to see that the minimum of limit of detection. So anything over and above the limit of detection, that needs an investigation. And we also have to review the previous products which have been produced in that campaign or the previous campaign, maybe previous three campaigns in the routine chain of equipments have to be checked, and it should be anything below LOD, okay, fine, but anything above LOD needs an investigation.
Thank you. Thank you very much. Mala, while you were answering this, somebody has posted a question for you, which is a new question. Uh, it's by Chetan. The question is rinse recovery is required. If yes, then should be performed as per coupon study. How to calculate acceptance criteria for rinse sample?
Well, so for the rinse and swab samples for both, you go by the macro limits what you have got, and you verify it. But for rinse and swab, both you need the recovery studies to be done, where you spike a known amount of sample and you check the recovery. As you do in the swab, you have to do the rinse also, recovery studies as part of your validation.
Thank you.
Yeah.
Thank you, Mala. And there is a question which is directed to you also, Thomas, just recently. During campaign cleaning and dedicated equipment cleaning, we discussed in day one session to test product residue. GTI, NSI, impurity and microbiology. But if the product itself met the required specification, is it required to test during cleaning?
So if we have these kind of campaigning, so we have the dedicated equipment, and there is a need to do a good risk assessment based on the fact that you would only have the same active, and David already mentioned before, we might see degradants or we have a microbiological issue with the dedicated equipment. Also, the campaign lengths can be really dedicated on these kind of factors to see how stable is the API, the active ingredient, and what kind of byproducts can exist if we have a very long campaign length. So is there a degradation? So these kind of circumstances is what you need to take into account. If you have a very safe process because you are just making an API, let's say, and you're doing crystallization using organic solvents with not a risk of microbiological issues and so on, then you might come away with that. But usually you would have to really demonstrate in a way that you are showing that you are not running into any risk by using a kind of analytics on the API, on the microbiological status, to really identify what is the campaign length I can use, and even how many batches can I continuously produce after each other without getting into trouble. And also here, the cleanability comes into fact, because maybe you have a buildup of residues so that you are getting really hard time to clean after a while, and then you might restrict your batch or campaign lengths to like 5 runs or 7 runs is what we usually see, to keep the level of residues in the equipment down to an acceptable limit, so that you can clean it in a reasonable time and also down to the limits you have calculated.
Thank you very much, Thomas. Thank you very much. And David, so I have given Mala and Thomas two questions without coming back to you because I have so many questions for you. I don't know where to start. Let me give you two of the questions which are directed to you. One question is, why elemental impurity not considered for cleaning validation?
Mhm.
Um, I need more information than that. What do you mean by that? Why
Uh,
element impurities not considered for cleaning
I have
validation?
just invited Raghuvendra on the stage to ask you this question in person because it seems to be it's incomplete. Raghu,
And
can you please accept the invitation?
can you give me an example? You should be looking at. So when you're doing your worst case, let me kind of give you a clarification here. So when you're doing your product characterization, you should be looking at all elements of your product, okay, characteristics and that, and including those would be any of those impurities that are associated with that. So when I do an extensive uh, a product characterization, I'm considering those as a part of my product characterizations, and if I my conclusion is that they're not worst case or they're not going to have an impact on my overall cleaning process, then I'm solely focusing on my worst case soiling. Okay,
Thank
if
you.
those before that, I'm going to be looking at and studying my degradants also. Okay, anything that may impact the product or the cleaning process and that. And that would give me more understanding. We do lab studies, we do design of experiment, and we challenge our material in the lab with different types of cleaning agents and that
Mhm.
to be able to evaluate which one's the most effective and which action is the most effective and that for cleaning. And from those studies, we transfer that into our cleaning development study. So we now identified our cleaning agents and that. Does that make sense?
Thank you. We already he just went off the stage again.
Okay.
Please mention in the chat if your question is answered. Can you please
I just
do that?
want to add one thing also on that, because if we talk about elemental impurities, this is very often related to catalysts and API production. So here
Yeah,
we really have to take care of that, but this is not a part of the cleaning validation, as we are making the cleaning validation to show that the API is cleaned down to an acceptable level. And the
exactly.
elemental impurity is something of the synthesis steps and where you have the purification of the API, and this is not really a focus of the cleaning validation itself.
Exactly.
Yeah.
And you want to focus on that. That's why you do your product characterization to analyze your worst case soiling and that. You still need to justify what the rationale is for your worst case and that. So that's why we don't focus on it. Thank you,
Uh,
Tom.
I just want to add top a few more things.
Mhm.
As
Yeah.
rightly mentioned by David and Thomas, this is more of the elemental impurities is for the product. Okay, depending upon the MOCs of the equipment that you use and the catalyst which is added, intentionally added, or a byproduct of element that could be generated because of the chemical interactions. And when you're establishing that these elemental impurities are within the specifications for a given product, this is
Yeah.
not required to be tested as part of the cleaning, because cleaning is more to do with the previous product contamination and not to do with the impurities in the previous product, okay, which could get contributed. So I don't
If you
think so.
start doing that, you'll be in cleaning validation forever.
Yes.
You'll be in perpetual if you start trying to look at all that. I mean, I'll be more than welcome to help you do
Correct. Again,
job
nitrosamine.
security. There'll be job security for all of us, wouldn't it? Good question. Good question. No,
Yeah.
because I know other people struggle with that same question. So I'm glad
So,
somebody
David,
asked.
I have another question for you. During cleaning validation in a new block, for the first product cleaning validation, where next product is unknown, how to calculate MSSR?
I think I think I sent that. I kind of went over that with you guys already in
Yes.
that.
I was about to
So
say that. Yes.
yeah, so I went over that one in detail. So maybe this is a late question in that. Hey, so what I would propose is whoever sent this question, send it to me, and I'll show you some physical calculations in that, and then you'll really understand what needs to be done in that. Because when you're doing cleaning validation and you got one product and you're entering in another product that's unknown, you should know what the unknown product is. Okay, um, you shouldn't have no unknown variables. Okay, because you're putting your facility, your process equipment, and everything at risk, and you're changing dynamics of your cleaning process, as well as the other product. So if you're bringing something in, you got to know what you're bringing into your facility. So
Thank you very
I
much,
don't
David.
know what you're bringing in. You could be bringing in anything. So if it's unknown, you shouldn't be bringing it in. I guess that's the simplest answer I
More
can give.
specific, kind of question
Yeah.
is going to be required. What we can do is, my team, please share with Vamsi from Beatrice David's email address, so he can send much more proper particular data and information.
Maybe position the question in a little different way than that.
Yes,
So
yes.
then that way I can answer
So
it
we can
good.
share your email with Vamsi,
Yeah.
and he can just
Yeah.
direct that question to
Because
you.
yeah. Okay. Good.
Right. Okay. So the question for Thomas that we have is: would performing a risk assessment on cleaning already during product development make sense? So this is a question that was given to us after your session because we couldn't
Mhm.
accommodate Q&As.
I would say absolutely it makes sense. And I can tell you from my experience that very often the R&D department develops new formulations, and they are in upscaling very easy with the cleaning because they say we will just use solvents, and they can get rid of a lot of molecules just using it. And then if you're going into the large scale manufacturing, you try to start to evaluate what cleaning works best. So we always outline that during the development, it is very good to have a look at the cleaning itself. And even what I always recommend is just have a look. What kind of cleanings are you performing today in the manufacturing site? And would this be also applicable for the new product? Because that can help you even in the grouping strategy that you say, oh, I see now this is also acid soluble residue and I have an acid group. Will it be a new worst case? I just have to look at solubility and toxicity and so on, but I already know where to put it. So these kind of things is really beneficial already during the development of a new product, or if you have a new product introduction, to look at this cleaning first of all, to have an idea where it stands in terms of your product portfolio, what you are producing today.
All right. Thank you very much. And Mala, I have a few new questions as well, which are directed for you. But before that, I would take this question, which has specifically come for you from in-person conference. The question for you is: manufacturing companies must check the cleaning agent absence and the drug substance in API.
Yeah.
Is Yeah.
Yes, we should do
Would they
this.
check
And
it? Is what they want to know. Yes.
hello.
Sorry. Yes. Yes.
Yes, we should do it. We can have a common method for the detergents. We can validate the method and check the detergents, because the same method which is validated for the product may not be suitable for the detergent. And for the
Yeah.
product, it can be separately validated and ensure that both are verified.
All right, all right. Thank you. And for David, I have a question for you, which is in three parts. Okay, so here we go. The part one of the question is: in API manufacturing, there are multiple steps of manufacturing, and the API molecule is being formed in the final step of manufacturing. These cases are generally handled by considering the limit for cleaning validation as 100 ppm. Uh, Clear till now? Then I'll put the second part of the question. Um, Okay, the second part of the question, same question, is how this 100 ppm might not be ideal, since based on the ADE PDE value, the limit may go lower than the limit derived from 100 ppm. And so, how do we handle such cases where the molecular structure of the intermediates is different from the API molecule, and there is no data available from clinical trials from ADE PDE for intermediate molecules of an API?
Okay, so I have some questions to go back. Is this a small molecule, large molecule? But this is an excellent question. Understandably, you cannot pick limits arbitrarily. The rule of thumb is not acceptable. I know some people think, hey, this is a rule of thumb, but the agency is going to challenge you where you got that from. Oh, I read a white paper. Well, how's that relevant to yours? Okay. So you need to have a scientific rationale. Yes, risk assessment is a great tool to use for, properly. However, if it drives the user to a non-biased conclusion, you can't. Your risk assessment. What I've found is sometimes risk assessments are used as a way of trying to mitigate getting out of certain situations. So let's take a different logical approach to that question. If you're performing a risk assessment on a product, you're looking at raw materials, API characteristics, and all those other things to determine the worst case molecule, right? Based on a criteria at each stage of the manufacturing. To tell you the truth, I don't really have an understanding of your equipment process, what molecules are common throughout the process, if there's product transformation, if there's common worst case molecules at each stage. Without understanding all those dynamics, I can only speculate. Okay, so there's more information that I would need to make a logical question. If not, then use toxicity for calculation. If you don't have that information, try using toxicity. I've given you today some really good examples of how to do different calculations to determine that particular requirements and that. If you look at those calculations, I guarantee you one of those would fit into what you need. Okay. But if the risk assess, now that being said, if the risk assessment results indicate that you have data to support this 100 ppm, then that is your limit. But disclaimer, you should be looking at the solubility, LD50s. At the end of the day, though, what you're really trying to achieve is looking at the API itself, right? That's what we're trying to get limits for. We're looking at the API itself, and that's what we're trying to achieve. But if you have intermediate acceptance criteria to show reduction in that, then you really need to be able to understand a lot more than that. And I don't think I have enough information to really say nay or yay. But if I have more information on your entire process, I could probably address the question a lot better. And you can email me and I probably would give you a really good answer. But that's the answer. With limited information, for me to say anything other than what I just said is speculation.
Thank you. Thank
Yeah.
you, David.
Well, maybe just to add to that, what I've seen that people are using then the kind of threshold of toxicological concern to get to a limit, but there you would really need also the knowledge if this molecule, for example, is not having a carcinogenic potential or not. So this is also then going into different discussions, because the TTC concept, so this threshold of toxicological concern, is different if you have a potential carcinogenic or if you have just a molecule what has not any of these kind of structures.
LD50, solubility, cleanability index, all those things are important to understand. And so exactly. You need to have all that. It's not just one thing. So do your product characterization and then come to your conclusion from a risk perspective. But I need more information. If I give you anything other than what I just said, I'd be guessing, and you don't want to be. You want real answers that we're going to help you out there. Does that make sense, guys?
Yeah.
Okay.
exactly.
And just to add on top it,
But I love that three-part question. It was great.
Yes, Mala.
yeah, so when you consider 100 ppm, it's nothing but 0.01%, or 10 ppm when we consider, it's 0.001%. So, considering that these molecules of intermediate or below stages do not have a PD value or an MDD to calculate the worst case approach, so I agree with both of you on the cleanability, etc. But so the least that can have as an impurity is also below 0.01% or 0.001%.
Mhm.
So that could be another reason for having those limits at a very early stage.
Thank
Hmm.
you. Thank you very much,
Whatever
Mala.
you choose, though, you got to have a rationale and
Yes,
justification
and a
for
risk
that.
assessment.
Yeah, exactly. Does
Thank you
that make
very
sense? Whoever asked that question, it's a good question.
Can
Just
you
need more
please
information.
give a thumbs up, Stanley, if your question is answered?
Yeah.
Yeah.
Okay.
Okay, great. So we have the thumbs up.
Well,
Okay.
oh,
So before I take the next question for Thomas, let me just make the announcement. Raghuvender, I know that you have a question for Mala. I am going to ask it. Please do not post the same question four times. I will ask that from her. Please hang on. Okay. Thomas, I have a question for you. How
Mhm.
elaborate does the cleaning have to be for cleaning during campaigning? What should be the timeline for cleaning frequency and testing? And is there a requirement to validate the process?
Yeah. So I think this is also going hand in hand with one of the previous questions. This is really something where you have to identify during a campaign what might happen to the product itself. So, is there a degradation of the API? Is there a microbiological risk? And if you can identify that there is no, or look also to the cleanability. So, how is the staining of the equipment? And yeah, you need to validate the cleaning in any way, even for dedicated equipment on the worst case situation. That means if you want to fix, like I can have a campaign length of 5 batches or 6 batches or 10 batches, whatever, then you might need to do this kind of scenario that you produce these batches. And David outlined, most probably you will take like 3 of these worst case runs if it's possible to identify then that the cleaning is working. But the condition is that you really make clear how you can set up the campaign lengths due to the stability of the API, that there are no degradant products, there's no risk of microbiological yeah, issues in the product, so that you can even verify that you might use 5 or 10 batches before the campaign ends and you need to do cleaning.
Thank you very much. Thank you, Thomas, for the answer. And so the next question that I have here is by Raghuvendra. Finally, your question is getting answered, Raghuvendra. It seems to be this was in the continuation of your last, statement that you made on the previous question. Can we use same worst case product of cleaning validation for DEHT study? Malavi.
It's hello.
Yes.
Yeah, it's S and no, because you should also look at the cleanability and the worst case with respect to the solubility. Okay, so one is what you take with respect to the MACO, because the MACO value, whatever you get the worst case, okay, the stringent criteria may not be the same thing for your solubility criteria. So based on these two, whichever is the worst, you can have that for your DHT cleaning, dirty whole time cleaning.
Thank you. Thank you, Mala. And I think, David, that you have covered this part in your PPT, but this question has come up on analytical testing is required after each campaign for product, which is having MSSR lower than VRL.
hmm. Yeah, that was already covered.
Yes.
I'm falling asleep here, by the way.
So Ankit was not paying attention.
Yeah, I think we already addressed this actually
Okay,
in
so I will share the PPT with Ankit. So if there is a answer in short, you can provide. Is it possible?
yes, absolutely. Go ahead. Yeah.
Thank you. Okay, so I will make sure that we share the PPT with him. And okay, so next question for David that we have is by Nimesh. Why ADE differ for the same molecule in different dosage form? Also seen different PDE by different organizations. How it can be harmonized and keep same similar as max daily dosage by entire pharma industry?
By I think the best way to do that is to join an organization that allows you to harmonize some of these, because I can tell you from my experience in walking into companies and that. Can you put the question back up? My experience is, yeah, is the question gone that fast?
Yes, I will
Yeah.
put it here again. Yeah.
So a lot of, so when you look at it, everybody takes a different approach to these, unfortunately. I don't know what Thomas and Ella, do you guys see the same thing, that, everybody sort of takes a different approach? There's no standardization. Um, but, we're trying to get there from the industry perspective. Uh, you know, Other than what I presented today as using as a tool in that from the ISPE, we tried to do as much as possible to harmonize these calculations in that. But I still see when I've gone into facilities, I just seen so many weird variations of this. And I'm going like, how did you ever come up with this calculation? You know?
Mhm. And
And sorry, go ahead, Thomas.
yeah, maybe just to add, so there is already in place an aid memoir from the PIGS, so that they have like this document PI 052 in June 2020. They even outlined that they know that there are different toxicologists looking
Yeah,
to interpret the adjustment factors and so on. And they have even outlined in this aid memoir that if you have a difference of a factor of 3, that is not that is normal and should not raise any concern. So if one has like identified a health-based exposure limit of 2 milligrams, then it also can be 6. So this is the normal threshold, that there are interpretations of the toxicologist by nature. But there should be an investigation if you are between threefold and a tenfold attention,
factors.
so
Yeah, yeah,
factors, yeah.
yeah.
And if you are more than 10 times, then this is generally not justifiable, and you should really look into if you are having the wrong starting point, if you have the clinical data. So that's really something where I would say this aid memoir of the PIC-S, so the Pharmaceutical Inspection Convention
That's where
Scheme.
the 5 factors come in to be able to evaluate that
Correct.
calculations. So
Mhm.
if you go back to the slide I showed, good point, Thomas. If you go back to the slides I showed where I
And
go over those five
I,
factors, yeah,
and I also feel it is dependent upon the purpose of use of the drug and the
yeah,
age factor, okay? Because the same drug can be given for multiple ailments and the concentration could be different.
yeah,
Okay, so the purpose of
exactly.
use is important, and the bioequivalent studies and the innovators. That is a genesis for a company to see what is the best, which can be processed. So the innovator study, data also helps on this, and different age groups. So that also wherein the PD can vary.
Thank you. Thank you very much, David. Thank you, Thomas and Mala. And some is the question that I have for you from Ankush from Collagens. How many batches shall be considered for campaign? Campaign study can be done on worst case or other than worst. So this is a question by Ankush.
Yeah. So I always get this question really dedicated to producers who are having identified a worst case product, but they are not making this very often. And then they ask how to deal with that. And if you have a campaign you really need to look what kind of circumstances you have, so what excipients you are using, and if you can show, for example, that you are using your excipients in the same way for your worst case product as for maybe the second worst case product, but you produce that more often, you might apply a rationale that you say, I will clean down to the level I have calculated for my worst case product, but I will do it with the second worst case because I do not manufacture my worst case that often. So this kind of situations is what we can see at some stages, that really worst case is identified, but I produce it once a year, and to run then maybe 5 validation batches in a campaign would be very, very time consuming, and you would have to verify every time when you do the cleaning, due to the fact that it's not validated already. So then really we can say, yeah, we will use the limits we calculated for the worst case, and we will analyze, but on the second worst case product, what is produced much more often.
Thank you. Thank you very much, Thomas. And Famala, for you, I have the question. If we consider cleanability for DEHT study, then how we can justify for impurity generation on holding dirty equipment?
Okay. So the T whole time study is a study which everybody knows that from the time you complete your processing up to the next stage of the cleaning when you start, that is a time period which you call it as a dirty whole time study. So when you do the studies, it is better you can compare even RRT wise impurity profile with respect to the last run which is done in that particular equipment. So that will give you some kind of insight as whether any trending of degradation impurities which are there, and also the bioburden, as Thomas already mentioned, that should be done, which give you some insight on whether the equipment is clean and dry enough for the next usage. Hope this answers your point.
Thank you very much. Can you please give a thumbs up if this answers your question? Okay.
Hey,
The ones who were
I,
listening, please give the
I do
answer.
hey, I do have one last thing to say about the product with the one with the MSSR lower than the VRL. For the analytical test required. It's obvious that there are many different compounds could not be really considered for evaluation by visual inspection, as the MSSRs based on the 1 in 1,000 or 10 ppms are below the VRL, or too close to it, so you can't be justified as a safe method for analyzing. Just to clarify that, that. Okay.
Thank you.
That was
Thank you.
one of the questions in that. So, yeah.
Okay. Thank you very much, David. So,
Makes sense? Up?
sorry?
I said, did that make sense? Thumbs up.
Yes. Yes. We just saw a
Yeah.
thumbs up as well. Can we do that again, guys?
Yeah. I want to
Great.
see some. I want to see some love in here. What's happening with those lovin's there? None of those.
We
Yeah,
can.
there you go.
Yes.
There we
Great.
go. We all feel better. See, everybody's smiling now.
Okay, so David, in continuation to your previous answer, Jagdish has asked you a question. What is alternative when method LOQ is higher than MSSR for high risk drug product?
That's a. I can you rephrase that? What is an alternative when the L?
So I think they are trying to ask if the LOQ of the method, analytical method, is higher than the visual inspection thing. How do we conclude this?
If the LOQ is higher than the visual inspection,
Mhm.
Then
So
how do
they can visualize the residues down to a level where even the limit of quantification for the analytical method is not achieving any result anymore. So that this analytical method is delivering a higher value as a limit of quantification than what you would see visually, so as a visual clean criteria.
That's a good point because we've. Let me give you. I think I already gave you an example of this. Okay, if you're if you visually see something on the surface and you swab it, and you're getting your results are meeting your acceptance criteria, does that mean it passes, or does that mean the visual inspection is accepted? Let me turn that question around. Okay, does that make sense? So I visually inspect something, I see residue on the surface, and then I swab it, and it meets my acceptance criteria. What do you do in that situation?
Thank you.
No, I'm
And
asking as a
oh,
question.
you're asking this question. Okay.
Yeah, I'm
Yes.
asking the panel, what do you do in that case? You reject it, right? You
Mhm.
reject the cleaning. It fails
Mhm.
because it failed on the visual inspection.
Mhm.
Okay, you can't. So so I'm trying to answer the question by proposing the question is that just because you have a good visual range and you're able to detect something and you sample and the sample criteria is met, what does that tell you about the sampling methodology? Okay, if it
Mhm.
still meets its acceptance criteria. Okay. So
Yeah.
that happens, by the way. That happens. So your clean and validation failed, right? Because it didn't meet your visual inspection. You might have met your acceptance criteria. Does that make sense?
And I think it's always very important to have the analytical method to be able to show you the maximum safe carryover limit of the API next to the visual inspection, so that
Yeah,
you really need the analytical method. Because if you even look to the Annex 15, it is outlining that visual inspection is a critical criteria, but also this cannot be the only method you are using to verify that the equipment is clean.
exactly.
Yes.
Exactly. So even if you visually inspect, you see residuals and you sample and the sampling method, your criteria was acceptance criteria is met, you still have to fail to clean it because it didn't meet the criteria for visual inspection. Okay.
Thank you. One question that I would like to ask all three of you is because I guess means everyone can have a different opinion on that. Anand has asked if you can suggest any good software for cleaning validation programs. If you're comfortable making one suggestion each one of you.
I've been using a couple of them. One that I like is the Bell Genesis Clean and Validation Program software. It's really good and robust. Um, I've used it quite frequently. So I don't know what anybody else's experience is,
Yeah,
but it
we are
works very
using
well.
we are using the Lucene platform called Clean with double E
Yeah.
in the middle. This is what we also partner with, so that is also quite
Mm,
sophisticated overall cleaning validation software.
mm.
Thank you. Mala, do you have any
I,
suggestion?
I agree with
And
Thomas.
by the way, Thomas, I use that too.
Okay.
I've used it in other sites
Okay,
um,
Okay.
and that.
Yes, Mala.
good to
So
hear.
That's a
yeah,
new
yeah.
that's a new product by you guys too, right,
Yeah,
Thomas?
Yes.
exactly.
Yeah,
Thank you,
yeah.
David. So Mala, you were saying so.
Yeah, yeah. So I agree with Thomas and David for clean software, but it's more known in the formulation. But for the API, it needs some more development. But
Yeah,
it's good to have that.
Yeah.
Thank
for
you.
final dosage forms, yes.
Yeah.
Thank you very much. So the question that I have for Thomas is what material shall be selected for establishing the VRL limit? If you
Haha,
could shed
yeah, the
light.
visual residue limit is usually done on stainless steel. And we really have always the discussion on what kind of level you need to verify that this material needs also to get like to a visual residue limit identification. Meaning, so if you have like a silicone hose, what you use next to the stainless steel vessel, should we implement also this visual residue limit for the silicone hose? And we usually do a kind of calculation. What is the portion of the total surface area in contact with the product, so the total product contact surface, and this kind of materials. We have these kind of rule of thumb at the moment. What we see is that if it is 5% or more of the total contact surface, a lot of people will tend to make this kind of inspection exercise, so to train the operators to make this kind of visual residue limit identification, so to make an in the laboratory them aware, oh yeah, we have this material like Teflon, it's a white plastic material, can we see the white powder, or can we see the product residues on them? But really there is not really as I know, not really a fixed threshold of that. And I have seen
Hey, hey,
one of our big customers going down now to 2% of the total inner surface, total product contact surface, to make this visual residue limit in inspections. I would maybe really emphasize the 5% is maybe a good rule, and even outline if there are some specific requirements, because also you might have like oily fatty substances what tend to stick on polymer materials much more, so to inspect a gasket, a sealing, what is maybe even a lower contact surface than 5%, but you know that that might cause a problem, so that this kind of situation should also be taken into account. So first
hey,
of all, maybe.
Thomas.
Hmm? Yeah.
Hey, Thomas, we're actually using Ecolab now to do some of our lab scale studies for us, and that
Hmm.
for two of my clients in the Bay Area, San Francisco Bay Area, and they're also selecting our detergents based on the material and that. So they're
Hmm.
doing all those DOEs for us. I just got a question to ask you regarding that. So I think when we do these actual visual inspections and that,
Hmm.
um, we try to apply like the 95% rule when it comes into the material construction, because if you have a large surface area and you're visually inspecting it and that, if it's mostly, it's in our industry, it's stainless steel and
Yeah.
that, right? So do you really want to waste your time on other materials that really you're going to have a low visibility, like you mentioned, silicone tubing and that? um, something that, so many uses you're going to throw out anyhow, correct? I mean,
Hmm.
they have their end of life. You can't be using them forever because they start turning yellow after a while, if anybody's been doing this, right?
Hmm.
So, when you guys do your study, do you calculate your view angle and viewing distance? Do you have like a sort of like a standard procedure that you use to do that?
Yeah.
And would you expect, you know, um, our audience here to have a standard procedures? I think you mentioned, someone mentioned between 215 lux for normal clean room light. That's typically what you see. But things like if you use one light source, you shouldn't use another, and that,
Yeah.
right? So that's important to identify. It's not only establishing that limit, but also the methodology for establishing that needs to be
Exactly.
standardized.
Well, and I think so. There are some recommendations how to do it. So how to spike surfaces. So we are recommending maybe to do a spot-wise approach, so that we use like a pipette and put spot-wise on the material of
Yeah,
construction the residues. But there are also other methodologies what are spreading the residue all over the plate, using, for example, solvents, these kind of things. But what we usually do is, yeah, the spot-wise approach, and then also to outline, you should have different distances to view onto the materials of constructions, maybe like 30 centimeters where you are very near in a manual cleaning situation, or like 4 meter 50, so that you stand away from it. Can you still see it on this kind of material? And this is something what we use to yeah, give as a guidance, but there is not really a fixed guidance document or something like that. It's something what we develop together with our customers to really make a kind of, yeah, what is sensible to do for the pharmaceutical manufacturing.
yeah. The
Thank you.
real true test of time is really being out in the field and using it, being able to do your visual inspections.
And lastly, what I would like to also add to what you mentioned is, so as you rightly mentioned, the angle, the distance, and the light is very important. But at the same time, when you do it in an analytical method validation, it's an instrumental technique that we are using. So the
Yeah,
sensitivity or the sensing is very much variant compared to what we do in a manual process of visual inspection. So
yeah.
that's another variability what you can detect for your limit of detection by an analytical technique. Maybe it could be visually seen or it may not be seen.
All
So
right.
maybe all put together we can have a risk assessment to say that what is the best way to have a limit for visual inspection.
Mhm.
Okay.
So it's not only looking at the API, but you're also looking at the detergent too, correct?
Yes.
Yeah.
Yeah.
So
Okay.
we have Meghal Shah who would like to access the stage. I'm going to hand over the mic to him. So he has a question which is with regards to the current conversation that you're having. So, Meghal, please accept the invite and you can unmute yourself. Yes.
Hello, am
Yes,
I
we can hear.
audible?
Yes.
Okay. I have. I want to ask one question regarding our conversation for LOD is higher than visual limit.
Mhm.
Where uh, uh, just I want to have a more clarity that if my macro value allows means that it is safe for the uh, carryover limit. So can I use that equipment if it is not visually clean because my macro value is higher than the visual limit?
So we were just talking about that, were we? Yeah, something like that. Yeah. Um, did you guys get that?
Yeah.
Um,
So, yes, did you get the answer to this? Because they were just discussing about it, Meghal, before you raised
yeah.
hand.
And And I think the answer is no. You cannot use a non-visual clean equipment to manufacture the next product, even if you know that you might have a dedicated equipment and so on. So the pre-requirement really is that the production equipment should be free of any visible residues before you start the next production.
Yeah, exactly. That's your criteria. Yeah, absolutely.
Thank you. And I've got this question already. Okay, I had a question for Mala. Let me just go back. Okay, yes. This one is answered. We are in continuation of that same conversation that we were having on the worst case scenario. So this is why I am just putting that question somewhere in between, but I've lost that question. Okay, let me give you the another question that I have for you in that case. I'll come back to that question again. Uh, just a second. Uh, there are citations that firm has not used worst case DEHT studies, which emphasize that we have to use same worst case. Is it so? So this is the question for you, Mala.
Yeah, I think we answered this. So what we said is, for the worst case, there are two criteria that can be applied. One is that a product where it is difficult to clean, okay, and the worst case in terms of solubility. So this also has to be given into consideration, the cleanability part. Apart from that, with respect to your macro also, there will be a worst case. So considering in these three categories, whichever is a worst, that will be good to consider in the chain of equipment.
Okay. Thank you
Yep.
very much. Thank you. And this question is directed to you, David. It's between same product cleaning, how much traces can be carry forwarded from batch one to batch two, and how to decide? Is by any
Well,
chance
have
is
you?
this? Yes.
Um, so, so what the question is between the same product cleaning, how much trace can be? So it's the same product, and you're asking how much trace can be carried over, how much residuals carried over. Is that the question? and can be carried over forward from batch to batch, and how to decide. Well, one, you have to look at your risk assessment to determine, what you decided as your original acceptance criteria for that. So what is your calculations? What are you using to evaluate? So if you're cleaning something and you're going into the next batch, what is the risk going into the next batch? Okay, what is your concern? Is it residual a product being carried over? If it's the same product, or are you concerned with microbial being carried over to the next product? What is your concern? You said trace. So all those things have to be still evaluated. You still have to reduce things. You don't want to be carrying. So you have to apply whatever acceptance criteria you establish for carryover of your product based on acceptance limits, and that you just can't say unclean it. So apply the acceptance criteria that you use. Does that make sense, guys?
Oh, guys, give
Thomas?
thumbs up. Okay,
Got it.
panel members.
I like the micro questions.
Thank you very much. Thank you, David. Thomas, the question that we have
I,
for you.
I find micro a lot more complex than that, because you're dealing with living organisms, and the agency loves to focus on microorganisms and that. I think these calculations for ADE and PDE are pretty straightforward in the industry, and that there's been a lot of harmonization in that discussion. Sorry, Thomas, I'm taking up your time by wandering on. Go ahead, you're up.
No problem. No
Batter
problem.
up.
I think
Batter up.
So, Thomas,
it's a
that
good ad.
question is for you.
Yeah.
That which agent is suitable for glass line reactor or to remove the stains?
Yeah, so here with glass-lined reactors, you really have to be careful, because if you take an too harsh chemistry, so meaning a too high pH, we know that these kind of glass-lined reactors getting corroded. So you have different materials, and to remove stains is really depending on what is the stain. If it's something like organic residues, very often a mild alkaline detergent can help you with that. If it's more like a mineralic stain, you should go for acid ones. And these glass lined reactors are usually not so much attacked by acidic solutions compared to alkaline. So strong alkaline is what I would try to avoid. But really the removal of the stain would be depending on the residue itself. If you need alkaline or acidic one, this can be identified very quickly in the lab. Just do a solubility trial and see where the residue is best soluble in. And so I can even give recommendations on different materials to be used on glass-lined reactors, what are not giving any corrosion.
Thank you. Thank you very much, Thomas.
I would like
And
to add
yes,
on this, slightly mentioned by Thomas, better to avoid strong alkali solutions. But after having an acid treatment, it is better to also check the pH of the solution, the final wash,
Mhm.
and then repeat the procedure of your product to product changeover. That will be good to, do, and then verify whether the stain is got removed or not, and
Yeah.
verify that sample as well.
thank you, Mala. Thank you very much. The question that has been directed to you by Ankush is, if molecule is UV inactive, then how to go for analytical method development and limit establishment?
Okay. So if it is in UV inactive, you can also look at RI detectors and verify RNIC. Okay. So this also can help you, because when you have UV inactive molecules, you cannot, do it by UV technique or HPLC technique. The other technique that you can use is TOC technique, which is one more technique which you can use it, or even with RI detectors, if it is possible, that also can be adopted.
Mhm.
Then
Thank
TOC, conductivity, RI, you can explore all these three techniques, and you can see what is the limit of your worst case is achieved or not.
And maybe
Thank
just to add on that,
Yes.
even the Annex 15 in Europe is outlining that if you do not find any specific analytical method for it, that you can use like TOC or conductivity as a parameter. And this is very often used for detergents because they do not have any UV active
Yeah,
molecule, and so the TOC, total organic carbon, is really good to be used and not like searching for an HPLC method. We have some customers even in India who said we really need an HPLC method, but for detergents it is usually not required as there is a low risk. They are not so toxic based on the health-based exposure limit calculation, and so like conductivity and TOC are the right methods to be used to verify the
that's a
detergents
good
themselves.
point, Thomas. That's a
Yeah.
good point. Yeah. So just think of that context and that. That's a good way of separating the two methodologies that you're using, because you can still reach your limit on acceptance criteria for your product carryover, but you could fail on your detergent level, and that's why you need a separate methodology to determine that.
Mhm.
Thank
Good
you.
point.
Thank you very much, Thomas, Mala, and David. David, the question that I have for you is: can we consider for microbiology content as macro calculation?
Not yes, I, but it's I wouldn't call it a macro. Okay, I would consider it a acceptance criteria for what's allowed to be carried over for bioburden level. Okay, and you can do it for endotoxins. I've done those calculations. For solid oral dosage, you can use the USP requirements and back calculate to get the acceptance criteria. And I do an whole chapter for sterile products. It's a different acceptance criteria, but you can do the entire surface area, calculate, assuming that the microorganisms are spread evenly on the surface, and that you can do it for rinse and that. So I'm not telling you, if you email me, I can send you those calculations. Or you can go out and purchase the ISPE guidance document because it goes into real detail in that. Okay, so feel free to email me any of the attendees. I can write better than I can sometimes articulate the questions.
Okay. So, is it okay that if in the chat box we share your email address there,
Yeah,
because we were
as
sharing
long
it
as
with.
I'm not getting inundated with answering the same question. I don't mind. I don't mind answering the questions associated with specifically if I'm doing consultant work. It takes a lot of time, but if it's a simple, straightforward question, I don't mind answering it. I don't mind sharing knowledge.
Thank you. Thank you, David. Thank you.
You're very welcome.
Mansi, please requesting you to share David's email address as well on the chat box so everyone can have access to it and reach out to him for this. Guys, please do not spam him. It's a humble request. It's more to do with questions. So please
Three
stick
days later, Dave's still there, typing away, answering the questions.
to.
I do like interacting when I'm presenting in the audience. I feel that I do better representation if I have like a whiteboard and I show the calculations for that. That kind of drives things home. Sometimes I ask the attendees to bring some of their, what their acceptance limits are, and then reverse calculate to see if they are able to achieve what they say they are and how they derive from it.
Thank
And
you, David.
I do the workshops where I do give them examples and that, so
Okay.
they can go out and do those calculations and see if they did them correctly.
Thank you. Thank you.
You're
So I know that means you guys are sitting in different time zone, and Thomas is just starting the day, and it's very late for David, and Mala is already at work. So we were supposed to wrap up now. Okay, do we have a room for one more question from each one of you? Please let me know. Can we have just one?
welcome.
Okay. So I'll have one question for each one of you, one for Thomas, one for Mala, one for David, and then we'll conclude it and I'll send you the remaining questions on the email. So the
Okay.
question for you, Thomas, that I have is if one of my product is intended to pediatric, pediatric population, is it required to consider ADE value of the other product also being manufactured on same equipment chain based on pediatric dose in macro calculation?
No, so the route of administration is really something you need to take into account. Even if you are doing the risk assessment and the health-based exposure limit calculation, you really need to identify how
enjoyed this session, and I enjoyed Thomas and Mel.
Thank you very
Cheers.
much.
Have a nice
And
weekend, everyone.
so,
Yeah.
just one announcement, David will be speaking at our conference in the next year on cleaning validation, and we will be sharing the dates with you shortly. Thank you very much, guys. And I hope it is okay if we send you the remaining questions. There are not too many because we addressed a lot of questions, but there might be a few questions. I hope it is okay if I send it to you in the draft.
Yeah, that'll be
Yes.
perfect.
Okay.
Thank
Okay.
you, guys. Thank you very much.
Hey, by the way, I just wanted to say the participants' questions were really excellent.
Yeah.
You know, I think everybody that probably attend and the questions probably help them with their other problems they may have too. You know,
They
so
are really liking your comment because I can see a lot of
oh,
hearts.
good. They were great questions. I really enjoyed them.
And
So
practical questions too.
yeah,
Very
absolutely,
impressive.
absolutely.
Thank you,
Okay,
guys.
okay. Have a good evening, everyone, or
Right.
should I say, have a good morning, everybody, or wherever you are.
Yes.
But have a great day. Thank you so much, and
Have a
cheers.
good
Bye
evening.
bye.
Have a good
See
Yeah,
morning.
you.
bye.
Bye.
Yeah, take care.